My dear readers, listeners, and of course, friends. Today I wish to delve into a fascinating corner of evolutionary biology that has captured the attention of researchers and enthusiasts alike. The impact of alternative splicing on the neo-functionalization of the homeobox gene family in the rapid diversification of the Hawaiian honeycreeper birds, known scientifically as the Drepanididae. The Hawaiian honeycreepers are a remarkable example of adaptive radiation, having evolved from a single ancestral species to occupy a wide range of ecological niches on the Hawaiian Islands. This rapid diversification has been the subject of extensive research, with scientists seeking to unravel the genetic and molecular mechanisms that have driven this evolutionary process. One of the key factors that has emerged as a potential driver of this diversification is the role of alternative splicing in the neo-functionalization of the homeobox gene family. Homeobox genes, for those not familiar with the term, are a group of genes that play a crucial role in the regulation of embryonic development and the determination of body plan in animals. They are characterized by the presence of a highly conserved DNA sequence known as the homeobox, which encodes a protein domain that binds to specific DNA sequences and regulates the expression of other genes. In the case of the Hawaiian honeycreepers, researchers have discovered that alternative splicing of the homeobox genes has played a significant role in the evolution of new morphological and behavioral traits. Alternative splicing, for those who may not be familiar with the concept, is a process by which a single gene can give rise to multiple different protein isoforms through the selective inclusion or exclusion of different exons during the RNA splicing process. A groundbreaking study by Liang and colleagues, published in the prestigious journal Nature Communications, shed light on this fascinating phenomenon. The researchers used comparative transcriptomics to analyze the expression patterns of homeobox genes in the brains of several different species of Hawaiian honeycreepers, including the Maui parrotbill, the Hawaii amakihi, and the iiwi. What they found was truly remarkable. The researchers discovered that alternative splicing of the homeobox genes was much more prevalent in the Hawaiian honeycreepers than in their mainland relatives, the North American rosefinches. They identified several novel isoforms of the homeobox genes that were specific to the Hawaiian honeycreepers, and showed that these isoforms were differentially expressed in different regions of the brain. Furthermore, the researchers found that the expression patterns of these isoforms were correlated with specific behavioral and morphological traits in the different species of honeycreepers. For example, the Maui parrotbill, which has a distinctive hooked beak adapted for foraging on insects and larvae in dead wood, showed high expression of a specific isoform of the homeobox gene HOXD10 in the regions of the brain associated with beak development and feeding behavior. These findings suggest that alternative splicing of the homeobox genes has played a crucial role in the rapid diversification of the Hawaiian honeycreepers by allowing for the evolution of new morphological and behavioral traits. By producing novel protein isoforms with altered functions, alternative splicing has provided a mechanism for the neo-functionalization of these genes, allowing them to take on new roles in the regulation of development and behavior. But the story does not end there. The researchers also found evidence for positive selection acting on the alternatively spliced regions of the homeobox genes, suggesting that these novel isoforms have been subject to adaptive evolution in response to the unique ecological challenges faced by the Hawaiian honeycreepers. This is a truly remarkable finding, as it suggests that alternative splicing has not only provided a mechanism for the generation of novel genetic variation, but has also been subject to the guiding hand of natural selection in shaping the evolution of these birds. It is worth noting that this study is just one example of the many ways in which alternative splicing has been implicated in the evolution of new traits and functions in animals. Similar studies in other species, from fruit flies to humans, have shown that alternative splicing is a pervasive and powerful force in shaping the evolution of gene function and the generation of biological diversity. But what are the broader implications of these findings? For one, they highlight the incredible power of comparative genomics and transcriptomics in unraveling the complex evolutionary processes that have shaped the diversity of life on Earth. By comparing the genomes and transcriptomes of different species, researchers are able to gain insights into the molecular mechanisms that have driven the evolution of new traits and functions over millions of years of evolutionary history. Furthermore, these findings underscore the importance of considering the role of regulatory variation in the evolution of new traits and functions. While much of the focus in evolutionary biology has traditionally been on changes in protein coding sequences, it is becoming increasingly clear that changes in gene regulation, including alternative splicing, can play a major role in shaping the evolution of new phenotypes. Finally, the story of the Hawaiian honeycreepers and their homobox genes reminds us of the incredible power of evolution to generate novelty and diversity in the natural world. Through the simple process of mutation, selection, and drift, evolution has given rise to an astonishing array of forms and functions, from the intricate dances of the birds of paradise to the mind-boggling complexity of the human brain. As we continue to explore the mysteries of the natural world, studies like this one will undoubtedly continue to shed light on the intricate and fascinating processes that have shaped the evolution of life on Earth. And who knows what other secrets may be waiting to be uncovered in the genomes and transcriptomes of the countless species that share this planet with us. But one thing is certain. The story of evolution is far from over, and the Hawaiian honeycreepers and their homeobox genes are just one small chapter in a grand and ever-unfolding tale of life, diversity, and the endless creativity of nature. Thank you for joining me on this journey of discovery, and until next time, keep questioning, keep exploring, and never stop marveling at the wonders of the natural world.